Hey Team Bio, welcome to your screencast on energy coordinate diagrams. I'm sorry that the last screencast was so long. I usually shoot for 15 minutes and I overshot a little bit last time. I'm going to try to make it speedier today. Okay, energy coordinate diagrams. We are going to be discussing how reactions work and how we can visualize them in a graph. And that's going to be important because our whole unit, biochemistry, is all about the atoms that make up organic compounds, that make up all the structures in our body, and then how those structures are involved in the biological reactions that make life possible. Okay, so brief reminder. Proteins. The monomer of proteins are amino acids. This pen is way too big. Whoop. Style. Oh, yeah. Okay, I'm going to shrink this down a little bit. Okay. The monomer of proteins are amino acids. When we string a bunch of amino acids together and we join them with peptide bonds, which is just a fancy way of saying the covalent bonds that hold amino acids together. We're going to form peptide bonds in class next week. So when we form peptide bonds, we get a polypeptide. And then a polypeptide itself is folded into a complicated three-dimensional shape to become a functional protein. And that relates to the topic of today because we're going to be discussing what enzymes, how they function in biological systems. Enzymes are biological catalysts. And most enzymes in the body, in the human body, and in all organisms are proteins, although some enzymes are RNAs. So what is a biological catalyst? It speeds up the rate of a reaction. And it does this by lowering the activation energy. Oops, activation, A-C-T-I-V-A-T, activation energy. And we're going to discuss in a second, if you have questions about activation energy, don't worry, we're going to define it in a second. I forgot the G. And they are not consumed in the reaction. So the same enzyme can catalyze multiple reactions again and again and again and again, as long as it has what's called its substrate. Okay, brief reminder about reactions. Reactions, generally we set them up and we say we have reactants on one side of our equation, and then we have the products on the other side of the equation. Today we're going to be talking about two super important biological reactions: cellular respiration and photosynthesis. I guess we'll start with photosynthesis. So photosynthesis is the building up, or I guess we should say plants take in CO2 and water and energy, and this energy is from the sun, and they create glucose, which you just built in class the other day, and oxygen. And then we could take this exact same equation, just copy it, and paste it, and then take our reactants and products and flip them, and then erase from the sun and write for cellular respiration. So cellular respiration breaks down glucose, it combusts glucose with oxygen to form CO2, water, and then releases energy, which is stored in the form of ATP. ATP is adenosine triphosphate, and it's the energetic currency of the cell. It's how the cell stores energy for very short-term use and then spends it. Okay, so these two reactions are just inverses of one another. The reactants of photosynthesis are the products of cellular respiration, and the products of photosynthesis are the reactants of cellular respiration. Okay, in an energy coordinate diagram, there are two different broad types of reactions: exothermic reactions and endothermic reactions. Exothermic reactions, an example of an exothermic reaction would be cellular respiration, and it's when the reactants the energy of the reactants is greater than the energy of the products. So in this reaction, energy is released. In endothermic reactions, the reactant energy is less than the product energy. So energy is required in order to get this reaction to take place. Okay, so here we have our two different types of reactions, exothermic and endothermic. So up here we have on the y-axis the amount of energy, and then on the x-axis the time. And we always write the reactants here, and the products down here. So over here, we can do the same thing, but the opposite. Sorry for the copying and pasting. I know it's annoying to write it all out yourself. So in our reaction with cellular respiration, you can see that the initial energy of our reactants is greater than the final energy of our products. So energy is going to be released in this reaction. Okay, let's label a few different components of this graph. I think that it's pretty clear that the total amount of energy, or I guess the energy difference, I want to say, between my reactants and my products is measured right here. And this is called the enthalpy, or the enthalpy change. And we write it as delta H, and it's the amount of heat that is required, or I guess the energy difference in between the products and the reactants. And it can be either positive or negative. So in this case, it's negative, which means that heat is going to be released in this reaction rather than required as an input. But notice that there is this peak here. So the actual amount of energy required to start this reaction is more than the total amount of energy released in this reaction. This is what's called the activation energy. So the difference between the energy of the reactants and the energy amount required to get this reaction started is the activation energy. So you know, if you have glucose and water just sitting out at your table, it won't just spontaneously combust at the current temperature and pressure that it's in this room. But if you provide a little spark, it will burn. Just imagine all this glucose energy is locked up in a log. The log isn't just going to spontaneously combust, but if you provide a little energy in the form of the energy of activation, so you light a match, for example, then the reaction can get started and heat is going to be released. You're going to burn that log. Okay, so this is the activation energy. And the reason why any reaction has an activation energy is because it's always more stable for two or more atoms to be bonded together. So in order to break the bonds between these carbon atoms to each other, we're going to have to put in a little energy to break the bonds. But then when we form these new bonds between the carbon and oxygen here, energy is going to be released in that reaction. So breaking bonds it requires an input of energy, and making them always releases a little bit of energy, and it just depends on the type of bond that you're forming, whether or not it is lower or higher energy. So this is going to be a higher energy bond than this. But being in a bonded state is more stable than being a broken bond overall. So that's what the activation energy comes from. Okay, so then looking over here at our endothermic reaction, here we have a positive delta H. So we've got to put in energy overall in order to form this glucose. But we also still have an energy of activation. Here. Okay, so why are we diagramming this all out and also talking about enzymes? Well, we said before that enzymes are biological catalysts that speed up the rate of reaction by lowering the activation energy. So if our red line depicts the rate of reaction without an enzyme, then this purple line is going to depict our rate of reaction with an enzyme. Okay, and bear with me. This always is a little bit annoying because Notability always wants to autocorrect my lines. Okay, so an enzyme is going to lower the energy of activation like this. And as a result, it is going to speed up the rate of reaction. Oh, that wasn't so bad. This shouldn't dip lower than over here. Maybe I'll try it one more time. I'll try a line like this. Okay. So our rate of reaction is going to be sped up like that. So our total activation energy here has been lowered because we supplied an enzyme to this reaction, even though the delta H remains unchanged. And likewise, here, we'll see the same thing. I'll start it off here, I guess. The rate of the reaction is going to be sped up. Oh, come on. Because of the presence of the enzyme, which lowers the overall energy of activation, the amount of energy required in order to break those bonds. Okay, so very briefly, how do enzymes do this? It has to do with their shape, how they're folded. They are folded in a very specific way, and they create a space that the substrate or the reactant perfectly fits into and orients the two substrates here relative to each other such that the reaction is much more likely to take place. So it has to do with their shape, the way that they're functioning as catalysts. Okay, I think that is it. And I'll see you in class tomorrow.